My next speaker is Dr. Farid Khan. He is an award-winning scientist and Chief Scientific Officer, PharmaCure, and he will be talking about future LOT and drug innovations. Dr. Farid Khan.
Thank you very much, Chairman. Can everyone hear me? Well, assalamu alaikum, and thanks for my brothers and sisters at APS for inviting me here. What I want to talk about today is my hero. My hero is Alexander Fleming. He probably saved 1 billion of us already. I'm talking about very high impact innovations, and I'm sort of want to create incubators of the future. I've started one in Manchester. I've taken 500 medical students through them now. And what I want to do is the future medic has to have training in science. So all my medics either have a PhD or a master's as well, because there's a tide, there's a revolution happening. And today my talk will be about new medicines, faster development of medicines, and why aren't we doing this in developed countries, and very fast, cheap methods to find things which we're able to do actually, and drug adherence, and also about the future. Manchester, in particular, called the Internet of City. It's going to be a smart city, and I'll talk a little bit about that as well. So, the question: how can we find safe and cheaper drugs and fast track them, and with patient safety as a key function? Well, if you take aspirin, and indeed if you take Viagra, they have off-target effects. You know what happens with Viagra, but with aspirin, not only does it cure your headache, but it also thins blood. So all of you as physicians, you know that certain drugs actually have other effects. Those other effects are probably more interesting in some ways. So because they are bioactive, safe, and bioavailable, these drugs, which are normally used for one indication, can always be used for something else. So you can fast track them in terms of FDA approval. Okay, they have safety efficacy already, and I'll explain why that is a major advantage in drug development. Also, if we're having drugs, right, 50% of us don't take drugs. That's a massive amount. So if you're not going to take drugs, you're never going to get better. So how do we increase that? So there's some technology which I've invented, and now, alhamdulillah, it's taken to the next level. It's been invested in and taken into the NHS. So I'll be a little bit talking to you about that. And how about, we're also going to be talking about the IoT of things as well. So this is Internet of Things, not LOT, but IoT. So we're going to be going into this. So my two talks are about drug repurposing and the Internet of Things. Normally I give this in about half an hour, so I'm going to rush a little bit. So drug, how much does it cost? £1 billion roughly, from research all the way to get one drug, and takes about 12 years. Forget that, I don't want to do that. I want a drug within 3 or 4 years, and I want it fast-tracked. So how do we do this? And I'll tell you how to do that. First, you have phase 1, phase 2. If you've got a repurposed drug, i.e. if aspirin can be used for cancer, for example, you can get rid of 6 years here. That's a very important point. So you can fast track it and then take it forward. So there are drugs that you can fast forward because they have the safety issues already done. Non-adherence. This is actually an old figure. It's about 300 billion per year of wasted drugs and people not taking them. Massive, massive amounts. In the NHS, only 500 million per year is wasted. 3/4 of people don't even take them as prescribed. In trials, in those things which are called drug trials, about 32% of the cost, loss of patients, is because of non-adherence. A normal clinical trial phase 3 is 40 million. 1/3 of that is just wasted. Okay, these are just figures to talk about. So let's change the tune. I mean, if you're going to find a drug, what is the biggest killer in the world? In 2010 in Pakistan, there was a flood. 2 million people contracted malaria. Okay? The largest killer in the world, and per year on average 3 million deaths per year, majority under children under 12. There is nothing out there in terms of vaccines. There's So how do you fast track, if you're looking for a cure for something, how do you fast track that? Well, for example, here we have blood cells. You have an infected one in human blood. The way we do it, this is working with universities. Here what you have is a drug, which is artemisinin. This is the parasitemia, so this is the parasites itself. What you can identify by just using human blood and feeding the blood with these parasites, you can actually, using FACS, cell sorting, you can see actually what are infected and what are not infected cells. Then all you have to do in those wells is add the drug, because it's in an in vivo environment. You add the drugs, this is what I did with Salford University in Manchester, and then you screen known drugs. There's only 30,000 drugs which is used on human beings. So why don't we just get them all, put them in wells, add it to the infected cells, and find which one works. So these are normal drugs that everybody uses. So then you can fast track. You've got a cure. And this is a paper which I want to cite next. So we did a screen. This is the number of parasites, yeah? Number 66 drug kills everything. And those are quite interesting as well, those two drugs. Then what we did with that 166, we did a dosage curve, and to find that absolutely it kills not only the multinuclear types of parasites, but also the mononuclear types as well. So this, from nothing, we've got a repurposed drug that commonly is used, and this is the paper that you should look at. It's the most cited paper in repositioning, and we found a drug called dry hydrochloride hydrate, which is a common used drug, and indeed, I released this because I had the power to do this to the world without a patent, so we can quickly get this in order. So please read this, and it shows a methodology, how to quickly find drugs for something which, there isn't anything per se. So let's switch that. So that was just a methodology to give you an exercise of how to do that. That was less than £100,000. And look how much we spend in charities. Why aren't we doing that bit of research? Why don't we fundamentally change ourselves and do this? Because knowledge is very, very easy to do these kind of things. Because I used to work for a drug company, and I know how to do this, and I now teach all my students this. So in the Western world, dementia, this is the next big thing. Only last week, official national statistics says this has just taken over as the largest killer in the UK now, from heart disease to everything else. So that's interesting. So you've got the normal brain here, Alzheimer's, you've got early sort of prodromal AD here. This is where you've got amyloid fibers. You produce these plaques, and these plaques then have an anti-inflammatory response in a later stage AD. I'm just going to go through this quite quickly because I don't know much of the time. But if you had drugs which can disaggregate this, so dissolve those plaques, that's quite interesting. Not only that, if you have an anti-inflammatory, that's also very interesting because that will block or slow down Alzheimer's per se. This drug here, which I can't tell you too much about, but it's a repurposed non-steroidal anti-inflammatory drug, which is commonly used. In our company, what we've done, just using brain cells, and these cells here, these is A-beta production, where you produce these amyloid fibers. Just by titration this, what we find that in neurological cells, drug 1 significantly reduces A-beta levels. So it's a novel mechanism of action. So it acts as a prophylactic. So this is very, very exciting results. But in addition to that, there's two other drugs that we're working on. One is a prescribed antipsychotic used in schizophrenia. Another one is an antidepressant. Those two, we did in mice. These are the amounts of A-beta here. Okay, these are control mice which has been accelerated to create A-beta in the brain to create the same sort of morphology in brains as humans in terms of the plaques. And then if you treat them with drug, you can see a huge difference here. So these are all different mice treated in this sense. So again, this idea that we can actually dissolve some of these plaques is interesting. But I don't care about mice, and I don't care about cells. I care about human beings. 950 to date drugs have been tested. Each clinical trial, about $100 million. So do the maths. All have failed. we need to test them in humans. What we've done in particular, because we're allowed to do in rare diseases, this is GSSS, which is German Strauss-Schindler disease. It's an amyloid plaques in the brain gets accelerated in this. We gave combination of drug 1 and 2. This female patient did not suffer any side effects. Within 3 days, her condition improved that she could interact with people. She couldn't interact before. She could watch TV. She could understand television. And so after 2 weeks, she could speak to some degree. She couldn't speak before. And after 6 to 12 months, her condition was clinically superior. And of course, we had to take her off the drug, for ethical reasons. After a while, she died. But the fact is, it completely changed. And so this is, again, what we're trying to do is accelerate that serendipity. I'm going to switch to medication adherence. This is my company, which now is off and raised this huge amounts of funding. This here is now, what happens to patients if they're not taking their drugs. This is a bottle that's given to clinical trial patients, and it reminds them on their phone to take their drugs. Drugs come out of the bottle, and you can log in, and right now we can see thousands of people, where they are, where they live, and they give them reminder systems so that they can take their drugs. If they don't take their drugs, they're never going to get better. It shortens clinical trials. How much? Okay, so then I'm just going to finish on this. So what we created, if this works, let's try this. Oops. So what we created is a whole Internet of Things system throughout Manchester that we're monitoring people, getting them adhered to their medication, and encouraging that. We also got a position, we're working with the University of Surrey over what we call a test bed to roll this out in dementia patients as well throughout the NHS. That's the scheme. So all of this data can go through there. We're working with asthmatics as well, so we can monitor them in real time and again, get that data and encourage them through their phones to make sure that they are actually taking their medication. And I will end with this, which is CityVerve Manchester. We have got a private partnership with Manchester itself, which has now created this thing called CityVerve, where we're monitoring huge amounts of patients, and in particular, management of chronic respiratory conditions, community wellness, and smart air quality. And I'll end with that. Thank you for your attention.
Thank you so much.
Sure.
Actually, Dr. Khan, you have, so far the methodology you have used a single molecule approach, okay? What my point is that in order to prove the productivity and outcome, nowadays the research is moving into the high density approach. So like a proteomic, like a proteomic, metabolomic, and genomics. I just would like to know that with that kind of like a 20,000, 1 million cell in one slice you can see in a robotic manner. Have you thought about that to use those for the drug effect?
I had a department run that. For the last 10 years, all pharmaceutical companies have done it wrong. They admit it now. Forget their approach. There's a thing called the phenotypic approach. The phenotypic approach is if you add a drug and something disappears, which is the phenotype, just use it. Don't care about the proteome. We don't care about what happens in between. The mechanistic action you can decipher. What you're looking for is a fast cure. And I've written a whole book chapter around this, which is now it's how to fast track that. For less than £100,000, you can actually do that. What do you need? You need 30,000 libraries. You get a couple of students to titrate that. You have an assay. You've done, finished. And why aren't we doing that? Do you understand? Yeah. It's just my frustration because I've started doing this. Now I've got 6 examples. Malaria is one. Alzheimer's, we just found some repurposed antibiotics, which is another big problem. But we can do this for many, many diseases. And I think, and the thing is that you can patent these off pathway for other diseases as well. So you can actually create enterprises out of these.
So it means you want a typically based approach.
That's absolutely right.
There's one question from the back.
I came from there too. That's my background. That's right.
Especially knowing the regulations are so tight, and
Okay.
you're dealing with patient lives, and how
Yes.
your fast-tracking medication is probably wouldn't really fall very well in the hands of the regulator. So I'm intrigued to know what your thinking is
Okay.
on this and how you go about this.
So remember that all of these drugs are already FDA approved for safety for a different indication.
Right.
Okay, so let's take aspirin, and you found it for cancer. It's already safe to use. So you've got off the hurdle. So my first sort of thing was how to fast track it. So the approvals for safety of the medicine is you have to find out whether the efficacy is good and whether you need a slightly different dosage. That's why you trim about 4
So
to
you're
5
talking
years
about generics then? You're not really talking about development or anything new here?
No,
You're
no,
talking about
generics. Yeah, so prescribed drugs which are already available, that are
Right.
about 30,000 drugs already available. What you're doing is you're repurposing. So, one drug can have off-pathway effects. So you can still patent for that other pathway or that other disease. And that's how you fast track it. So this is not normal. This is what pharma companies have been doing for the last 20 years.
Okay, all right.
Yeah.
My second question was, you talk about the IT and how, I'm very much into digital health and, in how we can connect with patients. And I'm working in that area. I see that you're doing a lot of IT here. One of the good things with obviously developed countries is you've got the IT infrastructure, so you can do what you've just shared with us. What I'm trying to understand is how do you ensure cybersecurity of, confidentiality of one's information? That's a big thing, especially when we're talking about patients and their privacy and everything else. How are you ensuring all of this with all that data you're collating?
So we work with the NHS. They have their own cybersecurity portal, so we work with them. So that's on the NHS side. On the private side, there's a whole cybersecurity company that we're working on, and I can't talk about that, but that's another talk that we can discuss.
Wonderful, thank you.
You're welcome.
Thank you so much.
Thank you.
You so much.